Thank you. Hi, everybody. There we go. All right. This is going to be a challenge if we're all going to be quiet. So my name's Neil Lamb, and we're going to make this really informal. So stop, ask, interrupt me, ask questions, ask me to back something up, to dig into it in more detail. I promise I will get to my career, because I have a pretty alternate career. I have a PhD in human genetics, but I lead an education initiative that creates hands-on materials and educator training and public outreach really all over the state, and is actually now expanding across the country. It's a pretty different approach than what I started out in graduate school. But I'm actually going to walk you through my career path and some of the decisions that I had to make and some of the opportunities that I was faced with, and maybe some of that will resonate with you. You may be an individual that knows exactly what you want to do. You're going to come in, you're going to get this training, you're going to this career, and this is the path that you're going to take. That may not be at all where you find yourself. And you may be grappling with, I really like this, but I also really like this. And I'm not so sure what kind of field I'm ultimately going to end up in and what opportunities are available. So I'm going to walk you through. I'm going to actually unpack the box of me and maybe give you a little bit of background. And you may be thinking yourself, what does your box look like as well? So the most logical place for me to start is to tell you a little bit about myself. Can you guys hear me? Is the volume okay? All right. The volume. I am the son of an engineer and a nurse, and my maternal grandfather was a PhD horticulturalist studying the environmental signals that cued plant development. So it's pretty much fated that I was going to end up in the STEM field. I mean, all of my birthday presents, Christmas gifts were science related. I had, the Science Kit 2000 when I was in late elementary school, where you could make all kinds of chemical concoctions. And at that point, before all the safety mechanisms, you could really blow things up. I actually set the kitchen on fire at one point. I had this engineering kit that I would wire things together. I had a plant cloning kit, and so I was actually, as like a 13-year-old, trying to clone, trying to propagate carrot cells in the kitchen, and I actually ended up with like a fungal infection in the culture, which got indicated to fumigate the kitchen. It was bad. But so here's a picture of my grandfather. His name also was Neil. He was Neil Stewart. He's my namesake, and we'll come back to that in just a second. But an important part of me is what my upbringing was like and the things that I was exposed to. When I was 10, I got my first calculator. That was a big deal back in 1980. And here's a picture of me with one of my science fair projects. I did all the science fair stuff. I think that my engineer dad secretly wanted to be a scientist, and so he lived some of his childhood back through me. But that was a really important part of who I was and what I was exposed to. And I say that for two reasons. One, you all have some sort of upbringing. It may not have involved whatever field you're in right now, or it may have been a really integral part of your life. Recognize that upbringing. You may not want to go down the road based on that upbringing, and that's okay as well. But the second piece of that is all of you are shaping the life of other people. Whether you have kids, nieces, nephews, people that are neighbors, you are also creating an environment that helps shape the early world of the next generation of students. Talk about what you do. Talk about the science. Talk about the impact of it. Talk about the challenges that you have. People are listening, and if we're ever going to get away from kids saying, the only careers that I know are what my mom and dad does, the doctor, the fireman, and the cashier at the grocery store, we have to do a better job of talking about what it is that we do. So that's kind of the early stuff that got me to this point. I got my undergraduate degree at Auburn. Auburn was frankly my last choice school. I wanted to go to Emrys. I had already said I was going to go to Emrys. I'd gotten accepted to Emrys, but I only got a partial scholarship to Emrys, and my parents were going to have to mortgage their house. And Auburn offered me a scholarship. And so I begrudgingly took it, and the first day I set foot on the campus was a week before classes started. Not the way I would recommend that you actually do that. But in hindsight, I got the most incredible education from that place that I so begrudgingly went to. It was a brand new major in molecular biology, so they gave me a huge exposure to coursework. I spent a summer doing an internship at Vanderbilt. I spent a summer doing another internship. The Vanderbilt internship was with diabetes. I did an internship at the University of Texas, San Antonio Health Science Center, looking at the immune system. I hated my Vanderbilt experience, and I apologize if there are any Vanderbilt grads in the room. It was not about Vanderbilt. I loved my San Antonio experience. I almost didn't do the San Antonio experience because the Vanderbilt experience was so bad. And my thesis, my advisor had to talk me into giving it another chance. I'm so glad he did, because I wouldn't be here if I had not done that other chance. So here's another piece for your box. Don't base all your decisions on if you like the one experience. It's so vast and so different. Whether you're thinking about a traditional or a non-traditional career, don't base every experience, don't base your decisions on one experience. Met the woman that would become my future wife at Auburn. I'm very grateful for that. That will become important in just a minute. You'll see why. And then I was looking at graduate schools, and I knew I wanted to go into human genetics. And lo and behold, I ended up at Emory. And the beauty of that was I got to go to the school I wanted to go to, and Emory paid for it. So that was spectacular. My parents really liked that. I don't know if you know, Emory, one of the largest endowments at Emory is from the Robert Woodruff family. And the Woodruff family is Coca-Cola, not cocaine, Coca-Cola, let me be real clear, money. And so the only thing you're going to find on an Emory campus are Coke machines. You will never find Pepsi on the Emory campus. And so in appreciation for all the money that went into my graduate career that I did not have to pay for, I am a lifelong only Coke drinker. There will not be a Pepsi in my hand unless we are absolutely at like desperate measures. So I got to graduate school at Emory, and the lab that I rotated through several human genetics labs, and the lab that I fell in love with was evenly split between doing work with Fragile X and work with chromosome non-disjunction and Down syndrome. And Emory was the hub in the early 90s of all the Fragile X work, because the discovery of the gene for Fragile X took place at Emory. It was the place you wanted to be. And a lot of the chromosome non-disjunction, the Down syndrome, was also taking place at Emory. And I was really lucky to be in a lab that covered both. And another graduate student and I, both in the same year, went into this lab. And my advisor, Stephanie Sherman, said, okay, I have a Down syndrome project and I have a Fragile X project. Which one do you want? And I said to my fellow graduate student, I said, you choose first. And she chose Fragile X, and I had the Down syndrome project, and that was one of the best projects of my life. It was amazing. Let me tell you why. I don't know how many of you know much about Down syndrome, chromosome trisomy 21. It's the most common form of mental impairment. Occurs about 1 in 600 in a live-born population. Some of you are wondering when I'm ever going to get to my career talk. I promise you, it's coming. I promise. Hang with me. We're going to thread all these Cheerios on this string. What do you know about risk factors for trisomy, for chromosome non-disjunction? Advanced maternal age. That's right. AMA. That a woman's risk of having a child, not only with Down syndrome, but of having a conception with any altered number of chromosomes, dramatically increases as she gets older. And at about the age of 35, it skyrockets. But we don't have any understanding of exactly why that is. So part of my project, and this is at a time, I don't know how many of you are in genetics, but this is back at the time when we were doing genetic testing, one marker at a time, using radioactivity. And so you had a Geiger counter clipped down near your gonads and another Geiger counter clipped near your heart, and you worked behind a really thick set of plexiglass, and you had a Geiger reader continually beeping at you. That was what the work was. But I walked into a project with hundreds of families of children with Down syndrome, mom and dad, and my task was determining if the non-disjunction error, so this error occurs in meiosis when egg and sperm cells are made, if it occurred in the first stage of meiosis or in the second stage of meiosis, and if something called recombination, which is when your chromosomes come together and exchange information in order for them to align properly, if recombination was involved. And I stepped into the right place at the right time with the right techniques, and we found one of the only molecular correlates with Down syndrome, which was really cool. So my first paper was a Nature Genetics paper, which was really spectacular. I mean, it was amazing. And it was not just a Nature Genetics paper, it was a Nature Genetics paper that somebody actually wrote like an editorial overview of, and there was work that was done in Drosophila at the same time that actually verified it. I mean, that's like when you fall to your knees and you thank the laboratory gods for the way that that all came out. The beautiful thing about that was that it came about, for me, purely by chance. I happened to say, you pick which project you want. And I ended up with that project. It was a great project. I worked with some incredible people. My mentor, Stephanie Sherman, taught me so much about what it means to be a scientist, but also what it means to have a balance of work and life. So she had her kids while I was in her lab, and every year she would take the entire month of August off, and they would pack up their kids and literally they would go backpacking across like Norway with the kids. They'd be gone for an entire month. It was an incredible experience in so many ways. And it was at the time when the Human Genome Project had really ramped up and people were sequencing, were beginning to dig through sequence, the technologies were beginning to change. Right at the same time, the world of science dramatically changed with the cloning of Dolly. Yes, this is a trading card of Dolly the cloned sheep. Some of you are really horrified that I have this, and there are a few of you secretly that actually really want the Dolly the sheep trading card. But it was a time when society was beginning to engage in questions about what does this mean? What does genetics, what's the power of genetics? What kind of opportunities are going to be available? Should I be concerned about it? And I fell in love. I don't know how many of you had to do this, but I had to have a teaching assistantship opportunity. I had to teach for one semester. And to be really honest with you, most of the faculty saw that as an enormous distraction from what my real purpose was, which was doing the science. And so I was encouraged to take the easiest, least work TA position. But ended up going a different route and TA'd in the medical genetics class that's taken by first-year medical students. And it was a lot of work, but I loved it. I found that I fell in love with talking to people about science. And then I got involved with an elementary school partnership project at Emory, and I was teaching in the elementary schools once a month talking about science and bringing hands-on activities in. Many of the people on my dissertation committee gave my mentor incredible amounts of grief about that, especially when I then signed up to TA the genetics course, the medical genetics course, another time. But to her great credit, she built space for me and pushed back against that. And at that point, I realized education has got to be a part of what I do, no matter where I end up. I loved the education. Now, let me tell you, when I was a graduate student, this is 92 to 97, so I'm quite old. Our graduate program only channeled students to the nirvana of teaching at a university as a full professor with an NIH R01 funded grant. That was the only path. And I was telling Rachel, right? I was telling Rachel that my second year, we finally convinced the faculty to let us bring somebody from industry on as a lunchtime speaker, and they put us in the smallest room they could, and there were 75 graduate students in this room for like 20 people, because we'd never heard anything about it. And as soon as he finished speaking and he was from Gillette. So why a guy from Gillette is talking to a group of graduate students, but we didn't care because it was our first introduction to something else out there. And then the faculty came in and told us all the reasons why that was an awful approach and we should never go down that road. So I'm hopeful that you don't encounter that. I mean, obviously, the fact that you're here and we're having this conversation must mean that there is at least some difference in the world, which I rejoice in. So the next chapter for me, there were like 5 papers that came out of this research, out of my dissertation. It was a combination of hands-on wet lab and a lot of statistics and computer and mathematical modeling. The American Society of Human Genetics every year chooses one graduate student that gets their outstanding predoctoral research award, and I was chosen to be that person. I don't tell you that to say, oh, look at me, wasn't that spectacular? But the next thing I'm going to tell you will put that piece helps put into context. I got my degree. I graduated and was hooded at the end of May. And the next week, I was working in a church. So, that was a dramatic departure. I left science. I totally left science. There are times in each of our lives where there are things that call at us. It may be doing something working in Nepal. It may be something that is working with underprivileged children. It may be Teach for America. For me, it was making a difference in the faith community that I was a really active part of. And I took a job as the director of communications and family programming at this really large Methodist church in the suburbs of Atlanta. The people at Emrys thought I had lost my mind. I had a faculty member suggest that maybe I needed a psychiatric consult. And I don't mean that to cast aspersions on them. This was a dramatically radical departure from this guy that had gotten these papers and had gotten this award, and there were people saying, hey, this place is a great spot for you to postdoc, and you can look at a faculty position moving down this track, and you should be doing this. And I said, yeah, well, that's great, but I'm going to go work in a church. There are times when you have to follow your heart, and you have to trust that even if you can't see where the journey is going to take you, that there is a path forward. Now, let me tell you what I did in this church. So when I got there, this church had about 2,000 members, and it was mostly baby boomers. And it was in this great neighborhood in Decatur. In 1997, the milkman still came to this neighborhood. And a lot of the people that had lived there and had built the community were aging out, and there were lots of young families moving into the community. And the church was really struggling with how to communicate with them. So in 1997, the newsletter was still put together essentially on a typewriter. Very blocky. All the programs were disjointed. There were people that, the Wednesday night family fellowship programs felt like a civic meeting, like a Rotary meeting, and they had things like boiled ham and pineapple casserole. So this was the newsletter announcing that I was coming on board. The people at Emrys thought I'd lost my mind. The people at the church weren't sure what to do with me either. They just couldn't quite figure this out. But I was really blessed to be in a place where I got to help shape what the structure of the church looked like and the programs to welcome new families. I was up as the liturgist in worship once a month, leading folks through prayers and through the liturgy. I was in charge of a Wednesday night program that we totally turned around and ended up with 500 people coming every Wednesday night. It was the hub of the community. I did a lot of teaching and a lot of talking about challenging issues of science and faith, which nobody had been willing to step into that avenue before. And it was an incredible time in our life because we lived a block and a half away. Our firstborn came into the world 3 months after I started this job, and my wife taught kindergarten at the Child Development Center that was there. So I had this idyllic 2-year experience that made me comfortable in front of large groups of people, helped me think about how to talk about difficult topics and meet people where they are instead of say, it's this or nothing, end of story. Helped me really find who I was. By the time we left, the newsletter looked at least a little bit more updated. Now remember, this is 1999, so don't, we actually got a website, which we hadn't had. And the church was a different place. And so about this time, I remember thinking to myself, okay, God, I think I've now done what you called me to do. Now what? Where do I go from here? Do I work my way up the church ecclesiastical ladder, not as a pastor, but do I move into district communications and the denominational? Where do I go? And I got a phone call from Stephanie Sherman, who was my advisor when I was at Emrys. We'd stayed in touch. And she said, let's have lunch. And at our lunch meeting, she said, would you ever consider coming back to science? I need somebody to manage my lab, and we're looking for someone to lead the medical genetics course for the first-year students and totally revamp the course and do almost all the teaching. Would that interest you at all? And after a lot of conversation, I finished up at the church, and I came back to academia. And that time in the church prepared me so well for that next stage. I was, after a year of managing Dr. Sherman's lab, the department chair came to me and said, we're about to start a high-throughput sequencing lab. High throughput at that point in time is very different than what high throughput is today, but at least it wasn't radioactive. And we'd like you to actually get that lab up and running and figure out the technology, and we have a whole bunch of projects with the medical school, research-based projects that we would like to flow through this genetics lab. Would you be willing to do that? And I said, sure, this was a really cool time. We had the sequence of the human genome. We were now beginning to really look at what the application was. So I then started a whole nother chapter where I ran a lab and managed a lab. And let me tell you, I sucked at it. I didn't have any management skills. No one had given me management skills. In 5 years, I went through 5 administrative assistants. It was bad. It was really bad. And I went to my department chair, who fortunately had not fired me at this point, and I said, you and I both know I'm a much better educator than I am a lab director. And I think if you are willing to create a position for me where all I do is education, with the medical school, the graduate school, the nursing school, the theology school, I'll go out to your potential donors and talk about genetics so they understand why what goes on in this department is so important. I think that will benefit everybody all the way around. And to his great credit, his name is Steve Warren, Steve agreed and built a position for me that was really atypical in the field. So here's another piece in my box. Be willing to take major risks. Be willing to create positions even if you don't see them at first. I got my foot in the door as the lab manager, widened it some with the teaching in the medical school, was willing to take a sidestep and run this lab, but then created an opening to be all education. You don't find a lot of people in science departments, academic departments, that are all education. So take those risks. And then there's a bunch of other work that comes with that. So I was working with the medical school. I was writing questions. Have any of you ever had to write questions, medical school style questions, USMLE questions? They are horrifying because you have to be able to answer the question just by looking at the stem, just by looking at the question part. All of your they have to all be multiple choice. All the multiple choice question options, the distractors, have to be the same length. They cannot use any of those clues that you always use when you look at a multiple choice question to figure out which one you think, which ones can I rule out. That one's way too short. Oh, that one's 5 times as long. That must be the answer. None of those things. And you can't have any none of the above. All of the above, none of it. It doesn't work that way. So I was writing questions for the medical school, and I got approached by a book company as saying, so this is Emrys Elements of Medical Genetics. It's a British medical book. And they said, would you be willing to write 300 medical school style questions that match all the chapters in this book, and we'd like to put them online. We will pay you, but would you be willing to do that? And I went to my department chair, and he said, you know, you got to do this outside of work time. I was like, yep, I get that. I went to my wife. At that point, we had 2 kids. And she said, well, okay, we'll figure out what that means. So I wrote these 300 questions. They went online. They had to be vetted through a whole group of physicians. That was an interesting experience in and of itself, because many of them got rewritten, and we argued about them. That went so well that the company came back to me and said, we'd like you to write a book for medical school students about genetics, a crash course book. That was cool. Now, this book probably has sold like 300 copies. I don't know. I mean, there's no royalty check for it. But this was an incredible experience. And, you know, it's really neat to be able to say I've written a book. But this took me in directions I never would have thought it would have done. I had to learn now how to write. I had to learn how to work with a team of editors. I had to be willing to take pretty serious criticism and adjust from that. And because of that, the authors of this book came to me and said, we'd like you to write some drafts of some early chapters. Now, you might notice that Jim Watson is the lead author on this book, the co-discoverer of DNA. And they said, we'd like you to put some drafts together, send them to us, and if we like them, then we'll contract with you to help write. So I wrote these drafts. They called me and said, we'd like you to fly out to meet the rest of the authors, and we'd like to put you in this, on Cold Spring Harbor, which is out in New York. It's this wonderful, idyllic place, for a weekend to meet with the other authors and to spend some time with Jim Watson. Okay. So I fly out. I have an afternoon with Jim Watson, which is a conversation we can talk about when we're not being recorded. It was an eye-opening conversation. If you know any of the things that he says, then that will not surprise you. But still, it was he and I in front of a fireplace on a windy March day, just the two of us. And that was really cool. One of the authors while I was there was a guy named Rick Myers. And Rick was the department chair at the time of genetics at Stanford and one of the leaders in the Human Genome Project. And his group sequenced about 10% of the human genome. He was one of the key authors. And he and I had never met before, and we started having a conversation. And he said, you know, it's really unusual that you come from a place that's so research-driven, but you do all education. What does that look like? What does that mean? Tell me about what your day looks like and the kind of audiences you interact with. So this is March of 2006. And so we started talking about that. And he says, you know, I've got some people that I would love for you to talk to. You've probably never heard of this group, but there's a group that's just been formed. It's called the Hudson Alpha Institute for Biotechnology. And at that point, I momentarily forgot who I was talking to, and I said, are you kidding me? I do know them. Now, here's where I'll digress. So I told you that I met my wife at Auburn. We got married. My wife's a Huntsville native, and her family lived in Huntsville. And in 2005, a gentleman named Jim Hudson got together with another gentleman, both entrepreneurs, Lonnie McMillan and Jim Hudson, with a vision to build a nonprofit research institute focused on genomics, with scientists in the building doing discoveries, understanding how changes in DNA impact health and disease and agriculture, and in the same building put biotech companies so that they interact and that the ideas of the researchers make their way over to products in the companies. They put together about $80 million in private money and another $60 million from the state. And in August of 2005, there was a huge announcement about it in the Huntsville papers. And so on that day, my mother-in-law and both of my brother-in-laws and every other member of my wife's family sent me newspaper articles, sent emails. My mother-in-law, I know you were able to read this article online, but I just wanted you to have a copy posted in front of you to refer to often over the next year or so. That's what you get for having a mother-in-law that loves you and wishes that you lived next door. So I had already read all about Hudson Alpha. My wife and I had said that as much as we loved Atlanta, we really didn't take advantage of life in a big city, and we would love to live in a place like Huntsville. So here, when Rick Myers is saying, have you ever heard of this place? I was like, yeah, I have. And we started talking, and for 2.5 hours we talked. And he said, you know, I've only just met you. We haven't talked a lot about this. Would you be willing to at least go and spend some time with the founders and talk a little bit about what you do? And you don't have to entertain taking a job there, but that might come up. You just might want to be prepared for that. So I gave him my resume. So I called my wife that night and said, you will not believe what has just happened. First of all, I spent 2 hours with Jim Watson, the co-founder, the co-discoverer of DNA, and I just talked to somebody who has a potential connection that would get us to Huntsville. And my wife's like, you have got to quit lying to me like that. And I was like, no, really. So I send my resume to Dr. Myers. He sends it to Jim Hudson. Jim calls me about 4 days later and says, can you come next week for an interview? Okay. So at this point, I have not told anyone at Emory that I am now driving to Huntsville to meet with someone that they have never heard of. And so Jim and Lonnie take me to the site where the building is going to be, and it is literally a giant mud pit. That's all it is. They're in the process of excavating it. So they walk me around, and then they bring me up to the office of the building next door, and they unveil this set of blueprints. And they walk me through the building, and what's going to be in the building, and the vision behind the building. And I said, where's your teaching lab? And Jim and Lonnie looked at each other and said, well, what would we do with the teaching lab? And so I started talking about the things you could do in education with students and with teachers and with interns and with trainees. And after about 7 or 8 minutes, Jim reaches across the table and puts his hand on mine and said, let's just cut to the chase. Can you be here in August? Okay, you don't know me, but I am generally a very thought-driven, pragmatic person. I build spreadsheets with pros and cons for everything. I assign numerical rating scales of risk and benefit for all these things. And here this guy says, can you be here? Now, there's no building. There's no scientists. There's no, here's the specific work we're going to do. There's no structure for anything. And I said, yes, I think we can. When the opportunity lands in front of you, don't be so afraid that you let it pass you by. I gave up an incredible job at Emory. I loved what I was doing at Emory. I was redesigning the medical school curriculum to take a job in a place that nobody had ever heard of, that had no scientists, that I could not even point them to a website about. And it was in Alabama. And I literally had some of those same scientists that thought I was crazy when I left to go to the church now said, are you even going to pack your shoes? Will you even wear shoes? Emory people are really nice, don't get me wrong. I'm painting a very bad picture. That is not the way I mean that. You know, there are great people at Emory. So I took this job. And in this job, so a lovely paperweight of the Institute. So just this great, spectacular 270,000 square foot building full of light and glass and water and trees, and it sits on 150 acres, and the centerpiece of the campus is a 1 kilometer walk and run trail that forms the shape of a DNA double helix in a park with 700 trees. I mean, it is unbelievable. So my job now, which gets us to what I actually do, so we've threaded the Cheerios now, I essentially have a twofold mission. I create or help create the next generation of the bioscience workforce. So I provide intern programs, middle school camps, high school short courses. I work with undergraduate and graduate lab courses and training, web-based activities. So I am helping students understand the career fields. And not just I'm working in a lab, but I'm in bioinformatics, or I'm in genetic counseling, or I'm in drug design. I love doing that. I love turning kids on to all the options that are in careers. They walk through the building and they see people with pink hair and they see people with tattoos and piercings, and that vision of everybody in a white lab coat with a pocket protector, I hear it shatter, and it's spectacular. And one of my favorite things is we've been at this long enough now that the kids that came to our middle school camps and our high school short course and summer academies have now gone to undergrad, have come back as interns, and now are getting jobs. That's cool. That's a big part of what I do. The other part of what I do is build a genetically or a genomically literate society. Nothing small there. Create a society that understands what genomics means, from the food they eat, to the tests they're offered in the doctor's office, to the medicines that they put in their mouths. So I work across the state. I actually left a meeting to come here that I'll go back to that is redesigning the entire 9th grade biology curriculum taken by every student in the state of Alabama. That puts more hands-on, rigorous, lab-based activities that takes us beyond know what the Human Genome Project did. That was the extent of what we were finding. That's it. Okay, can we please update this back from something that ended 15 years ago? We create kits. This is one of my favorites. So they go in these blue dot boxes. Middle and high school students really struggle with chromosome behavior during mitosis and meiosis. And when do chromosomes segregate, and meiosis 1 and meiosis 2 and all these pieces. So to help understand chromosomes, we made chromosocks. So these chromosocks come in different colors and sizes and have different alleles embroidered on them, so you can tell which one came from which parent. And kids actually, we went to an Alabama sock mill and made about 50,000 chromosocks. And we use these all over the state, and students, actually there's another bag so that when they replicate their DNA, they have to make a second copy. So right off the bat, you can see when you say replicate your DNA, did they just put the maternal and the paternal copy together and stick a rubber band around it, which is what a lot of them do. A lot of the teachers do that too. Or do they actually realize I need to take the other lowercase f copy and the other uppercase f copy? It's a great, we do all kinds of kits like this. One of my favorite ones is something called Disorder Detectives. There are 15 game boards. They're case studies. So this is actually a case study about a 40-year-old female who is pregnant with her 3rd child, and there was an atypical ultrasound. And so here's one copy in white on the board of your chromosomes, and then the other copies are in vinyl clings. So you actually can take them off, match, and put them where their match is. So this case, it's a little hard to tell, but we have 3 copies of trisomy 18. 3 copies of chromosome 18. So this is a trisomy 18 family. There are 15 of these. They're different. They're all color-coded so that a teacher at a glance can see what each student's working on. We identified these clings so that if they get dirty, if they get lost in the parking lot and somebody picks it up, you rinse it off and it works. And there's a whole student manual that goes along with it that teaches kids about first-person language and how to not say things like, that's a Down syndrome child, and instead say, that's a child with Down syndrome. It's a small piece, but it is a reframing of how you have conversations about people that are different and all the messages that go with that. And the students, when they do this activity, they actually take on the role of the head of a medical cytogenetics lab. And so they actually fill out a cytogenetics report that goes back to the referring physician. And then they actually have to give parents information about where they can go to learn more about what their child is affected with. So it covers lots of content. I have one of the best jobs in the world because I take the amazing, cool science that takes place at Hudson Alpha and around the world and turn it into activities that students and teachers and the public and lawmakers and judges and physicians get. That's what I get to do. We built an app called iCell that teaches kids the 3-dimensional structures of cells. We had 1 million downloads last month on iTunes. It's free. It's cool stuff. I've built an online, my team, I did not do this. My team has built an online serious game called Touching Triton that takes place in the context of a 20-year mission to a moon of Neptune. And there are 6 people that are chosen to be on this mission. And the students look at their medical records, some genetic information, and their family history and identify what diseases they're at highest risk of developing, including Parkinson's. And then they have to pack the ship collectively, because there's not enough room for everything. They have to pack the ship with enough stuff that everybody stays healthy, and if they get the disease, they're treated for the 20-year journey out and back. That's cool stuff. I love what I do. It's probably time for me to be quiet now. I've probably run long enough. But the point, I think the points that I would leave you with, every interaction is an opportunity for you. Every connection. My job at the church, my job as the director of the lab, my job as education, every single one of those gave me something that lets me do what I do well. It equipped me. So look for those opportunities. Be willing to take risks when your heart leads you somewhere, even when people say, that's crazy. Make sure that you've thought through all the consequences. Don't just leap blindly, but be willing to go on those paths. And recognize when feedback from other people is valuable and when feedback from other people is not. And be willing to swim against the flow. So, how much time do we have? Do I need to be quiet now? 3 minutes. So let's take questions then. Let me stop at that point. So that's my box. So, questions from you guys. Yeah, how do you build the skill of being able to talk to the general public about science? That is such a critical piece. Good analogies. Find the right analogy that people can understand. I use DNA as a recipe book all the time, and it carries me through in lots and lots of ways. So make it understandable. The way you talk to the public has to be the complete opposite of the way you talk to each other and to your colleagues. You are trained to be as specific as possible, and in fact, you're dinged if you use jargon, or if you are broad, or if you don't cover all of the little tiny pieces. Get rid of all of that when you talk to the public. Cover the broad categories. Recognize there's going to be all kinds of circumstances and pieces where that's not always true, but don't get in the weeds. Your audience will be lost and they will not appreciate the intricacies of that. Keep the main point your focus. And read your audience. If your audience, stop and have everybody stand up and take a stretch break. Throw a joke in there. Rewind and say, all right, so let me reiterate what we've just talked about. In the big picture, the reason why this is important is because. And be willing to say, I don't know. That is your best friend in many situations, and it does not indicate that you are stupid or that you don't know. It means there's a limit to my knowledge, and I'm not going to make up a story or tap dance around to try to give you an answer. Let's figure out how we get an answer. Those would be some of the key things I would tell you. Others? Yeah. In some cases, find the audience that you like and jump in and volunteer. I went to my local school and I liked really young kids and I liked high school kids. Middle school kids, not so much my cup of tea. And I said, would you like a scientist to come in and talk about some of the things that you're talking about in your class? And once they realized that I could communicate with them, so back to your point, and that I wasn't going to talk over the kids' heads, and that I was going to bring in fun activities, then they loved that. So that gave me some volunteer experience. Another part of science education is science communication and being able to write about science for general audiences or for specific sub-audiences. There are more opportunities there than you ever would imagine. Groups that are advocacy groups for kids with disabilities, for reducing pesticide use, for For any number of science-related things, they need somebody to write copy for them that is compelling, that makes sense. Volunteer for that. Get involved. Ask textbook companies if they need somebody to edit content. The Encyclopedia Britannica is always looking online for people to help write specific pieces of content. And you get named as an Encyclopedia Britannica author. So look for those opportunities. You don't necessarily have to go to a school system and become their science district coordinator. But start with whatever is immediately around you and then build that portfolio that way. Others. This was really atypical, I know that. This was probably not at all like the rest of your career talks. But I've got a really atypical kind of career and an atypical way to tell that story. How did my team, yeah, that's right. How did my team develop our kits? So we start by saying, is there a gap? Is there a need? And we vet our way through, is this what somebody just told me, or do I actually have evidence to back up that there's a real problem in understanding this concept? And once we've said, yes, there's a real problem, and we're going to step in and provide a solution, then there's a whole process we go through that says, what's the most effective way to bridge that gap? Is it a bunch of hands-on materials? Is it an online activity? Is it something that we need to retrain teachers? So what is that? How do we develop that solution? And that really requires a huge amount of legwork and background. And my team has expertise in digital and in clinical and in classroom, middle and high school. So I've got people that I can go to for that. And then it was, all right, how are we going to fund this? So the state of Alabama put some funding for us in their education budget. The Institute is a huge supporter of what I do. They provide $500,000 every year for me to do my education things. And we write a lot of grants. And we do a lot of private funding. And then it's gaining the confidence of the people that are your end user. So I need to put out a quality product that's understandable, that they see fills a need. I think one of the challenges is that scientists sometimes say, I had this great activity and I think it should go into a classroom. And they just take it to a teacher and say, here, put this in your classroom. Or they write it at a level that's way too complicated and they don't recognize how it fits into the broader picture. So it's really a multi-step process.
Just in prep for the next event, we'll go ahead and wrap up. If you have any questions or anything, feel free to come up and talk to them while we launch everything up.
All right. Thanks, everybody.